So welcome back, everybody. Thank you again to Jenny for an inspiring presentation. So it's my pleasure now to introduce our final guest speaker for today, Peter Nguyen. So Peter is a research scientist at the Wyss Institute of Biologically Inspired Engineering at Harvard University. In many ways, the Wyss represents sort of an aspiration for the HPBE in terms of the mix of different disciplines and the aspiration of the whole team. So I came across Peter's work actually in a review paper. In fact, I have a copy here. It will. This is a very we have a number of these in my house in various places with different markings and annotations on. So Peter was first author on I say a review, I would say the review on engineered living materials in advanced materials. It was the first paper that I'd read that brought together the field in such a systematic way. And for the first time, I could recognize what we were trying to do in the works that were cited and the way that it had conceptualized the field and brought it together. So I reached out to Peter and spoke to him. This paper has since become the first text that we cite for all of our students working in this field. So it's a rite of passage coming into my lab that you read the paper before you get into there. What was really fascinating about it is it not only dealt with the scientific principles in a remarkably readable way, given some of the complexity of the ideas that are behind it, but also reflected on some of the wider context in which this work is taking place, including artists and designers and ideas of scale up and so on. And then speaking to Peter later, we realized, I realized just how far your scope was in terms of depth and breadth, and some of the ideas that we had suddenly seemed not quite as radical as they had done first of all when we spoke to you. So needless to say, we had to have you here today, Peter, so thank you so much for joining us. I will invite you to share your screen. Afterwards we'll do a Q&A, and Josh will actually be joining me for the Q&A when we finish. So if you share your screen, Peter, and we'll get underway.
Sure, absolutely. Can you guys hear me?
We can hear you perfectly.
All right. So I just want to thank Martin and Meng for inviting me, and that was a very generous introduction. I'm glad the review has found utility, and I had a bit of a challenge trying to figure out what I wanted to talk about for this talk. And so I wanted to go through, I saw the rest of the talks for this particular session, and a lot of them were rightly so focused on from an engineering and an architectural perspective. And so I wanted to bring a different sort of background from a synthetic biology point of view. And I don't know how many of the people that are watching are synthetic biologists or that are familiar with synthetic biologists. So I think I wanted to start with what inspired me about living materials, not just from a research point of view, not just from an engineering point of view, from an architectural point of view, from an artist's point of view, but from all of these combined as just an engineering and kind of design principle in general. And so with that, I'm going to go through my own inspiration for living materials, where it kind of, I guess, was seeded from. I'm going to use the term seed a lot throughout this talk, and I feel like I keep gravitating towards seeds. And when I think of living materials and how seeds kind of have the nugget of information that can take in all the energy and matter in its surroundings and grow into something far beyond what you hold in the palm of your hand. And some of the inspiration from that is actually from my childhood. And so we'll go through two inspirational themes that kind of drill down to basic principles. And I feel like these inspirations, they are fantastic examples of inciting the imagination and what is possible with living materials. And inspiration is from one, the Fortress of Solitude. For those of you that don't know, that is Superman's retreat, basically, or his headquarters, and something called Grey Goo, which we'll talk about in a bit. And then we're going to go through two vignettes. And these two vignettes are kind of areas of focus in my own work. One is more in the past, which is biology for making materials. That's going to go through my more traditional work in the past that is focused on living materials. And the second vignette is materials coming from biology, materials containing biology. So you have an everyday material, and it has actually embedded in it biology that you can't see, that you don't even know, and it's working in the background, and you don't even notice that it's there. And so that is newer work that I haven't really talked about, and we just published a paper just last week on it, and I'll talk a little bit about that. But that kind of veers away from living materials into something else, which is biohybrid materials. And I'll go into why I was more interested in that in a bit. And then also I'll close with what I see are the challenges and some of the opportunities that are just explosive right now in terms of growth that I see for moving living materials from an academic exercise into something that's a bit more mainstream and everyday technology. That's something that I've really focused on in the past couple of years is bringing it out of the lab, because we can sit here and publish, academic papers till we die, and then, who says whether or not the technology that we develop will have any impact? I kind of would love to see this become real everyday technology in my lifetime. So I'll go into that next. So inspiration, let's start there, and let's kick it off. So this inspiration, I'm going to play you guys a clip from 1978's Superman movie. This is by director Richard Donner, who he actually passed away this past Monday. So this is dedicated to him and his ability to incite the imagination of kids, big and small. And so this is a clip. I'm just going to give you guys a quick background of what's going on. This is a young Superman, and he's traveled to the Arctic, frozen Arctic, and with him in his possession, he has a piece of his dead homeworld, Krypton. So the planet's exploded and gone, but he has a little shard from it, and instinctively he pitches it into the frozen wasteland, where it sinks into the ice. And from this, you get something that is like a living material. And as you can see, that little crystal, that little shard has a program in it to look at its surroundings, attain the necessary matter, and build a structure out of whatever it can find, basically in situ resource utilization. And in the end, he builds a structure, and that structure is a crystalline fortress made from that one tiny shard, okay? And so that encapsulates, to me, that really, this scene was ingrained in my head since I was a kid, since watching this on TV. And so to me, it really encapsulates, it's obviously a sci-fi trope. It's science fiction, it's not reality, but the concept of taking a shard of something and having it reproduce into something larger, something with more structure, with more complexity, with an engineered function. Here, it's a fortress or a structure that you can actually live in. That really captures the imagination. And it's not so far in the realm of fiction that it's not possible, because I can just say, hey, I took a seed and I tossed it into a field, and before I knew it, I came back and it's this gigantic tree that eventually would probably get cut down into timbers and made into a huge house, right? And we do all that. We have that magic that we don't even think about, and it's just everyday technology to us. But this really was my first inspiration um, in terms of living materials. And the principle that it embodies is this principle of self-assembly and self-organization. And this has obviously been studied for a very long time. It's something that a lot of scientists, researchers, designers, artists are interested in. And here I'm showing some different examples. On the left, you have BV reactions. So these are Belousov-Zhabotinsky reactions. These are chemical reactions. They self-organize into these self-oscillating patterns. And so what you see there is not an animation, that's an actual chemical reaction that's occurring. And you have these gradients, these redox gradients, so reduction oxidation gradients in these chemical reactions. And as they evolve, they form these repeating patterns that there's absolutely mesmerizing. And this is a great example of self-assembly and self-organization in a very simple system. This is a very simple chemical reaction. Anybody can do it. You just have to have the right chemical components. And also, we have self-assembly and self-organization in everyday technology already. If you have an OLED TV, if you have any kind of nanoparticle light-emitting diode, it's used in solar cells nowadays. They use self-assembly to basically build and fabricate nanomaterials that have very distinctive electronic properties that we need. And so this is a basic chemistry. Well, I would say it's more advanced chemistry, but it's nanochemistry at this point, where you're developing nanoparticles and you're building nanoparticles. And you can see on this very edge here, you guys can see that there are nanocrystals being assembled from different elements, and they have very different distinctive structures, and from that distinctive functions. And also we see self-assembly and self-organization in living systems as well, which is where we're going to veer into soon. And here on the right, you have a diagram of tobacco mosaic virus. This is a virus that infects tobacco plants, so it's a plant virus. But what you have is in red, you have a piece of RNA that forms a helix. And around that one helix, you have a single protein that wraps around it and forms a very ordered tube. And that is the virus itself. And so the virus, it's a very simple program made of 4 proteins. And one of the proteins knows it's evolved to wrap itself around the viruses. And this also happens almost in a crystalline formation as well. If you look at these under the TEM and you do crystallize these, they form these very nice rigid structures that almost look like crystals. All right, and so those are 2 examples that so that self-assembling, self-organization coming from that one example that I just talked about before. And in terms of living things, so we talked about crystals and how crystals can form from this one seed, right? Biology does this, and biology self-assembles itself through what I see as a kind of recursive self-assembly. All of us, every living thing usually goes through a process where you have a multicellular form, and then it goes back to a single cell, which you can think of as a seed for that particular life form. And that whole self-assembly process of going through a multicellular state and going back to a single cellular state is somewhat recursive. It goes through generations, unbroken through time to the beginning of life on Earth, right? So every single person that's listening to this today was a single cell, was a zygote at some point, and you began to accumulate energy and matter from your environment, and you use that to self-assemble the rest of you, essentially. And before you know it, many are already far ahead of me and have created more zygotes. So you've created offspring now, and the cycle has started anew, right? And we're just part of this cycle that recursively iterates through time, through generations. And you have this constant single cell drawing in energy, self-assembling from the environment, and making a different life form until you go back to a zygote, right? And so that's the power of biology that kind of amazes me. So the next inspiration comes from something I picked up in college. So I read this book in college. It's called The Engines of Creation. It's by a scientist called Eric Drexler. Okay, and basically in it he popularizes the notion of nanobots, nanorobots, the ability to fabricate things from self-assembly, so from the bottom up. And so this uses self-assembly, which is a physical manner, a physical way of assembling atoms. But the way he does this is through small robots that are made through nanotechnology. And as you can see here, this is a simulation of a gear. And this gear isn't made of like, a bulk metal that you make from top down fabrication. This is made from atom by atoms putting together a small gear. So it basically, a lot of this nanotechnology is taking what we have built in terms of engines, what we have mechanically built in everyday world, and kind of miniaturizing it down to the size of assemblages of atoms. And with that now, the idea is these nanobots now, if you shrink them down to that size, they can now assemble things atom by atom. And so that opens up a whole new field of fabrication where we're no longer tinkering with wrenches and things where, again, you're building from top down. You're building things molecule by molecule. You're building things atom by atom. And that really opens up the field to making all kinds of different materials that have never existed before, and also different methods of fabrication. And that's something we're going to go into in a bit. And so here, this image shows a nanorobot that's repairing a piece of DNA. So it's used in science fiction a lot. You see in science fiction, they use it. Somebody injects themselves with nanobots and it repairs all the damage in your body and gives you immortality, for example. That's a sci-fi trope that has been used over and over and over. And that's interesting because we already have machines that repair our own bodies. They're called enzymes. And that is something that I kind of realized back in college is, do we really need these fantastical nanobots that are completely synthetic and made of atoms, whereas nature has already made these nanobots for us? Can we utilize them instead? And the corollary of an assembler or a nanorobot that makes something or builds something, is a nanorobot that disassembles things. Right, and that's called the gray goose scenario. That's also introduced in Drexler's book. And the gray goose scenario is popularized to the point where howstuffworks.com needs to make a graphic about it, and it's about how the world will end. This is used again in science fiction a lot as an apocalyptic scenario of nanotechnology run amok, where you have these nanorobots that basically all their task is to do is to disassemble things and take something and basically take it apart into its atoms, and also create more of itself from the atoms. And so you have a nanobot that all it does is disassemble everything else around it and makes more of itself. It will eventually have exponential growth and disassemble basically everything that is made of matter, including the entire planet. And so the entire planet will just turn into this gray goo, which is full of these disassembling nanobots, right? So it's great, science fiction fodder. It's used a lot. And so, we also have things that disassemble in biology. The proteases, we have nucleases, and they've been evolved to protect us from other living things. So, you see the trajectory of biological evolution imprinted on this idea of nanotechnology in terms of its assembly and disassembly. And so the key concept here for the nanotechnology that I want to focus on is programmable molecular engineering. From the book that Drexler wrote, he has this passage that I want to read. And it's about, quote, growing a jet engine from a brew of tiny robots in solution. As you watch, the brew quickly morphs into a solid piece of complicated equipment. And so the idea here is to have something incredibly complex that we build right now from top down, You throw it in a vat full of these nanobots, and the nanobots will take the atoms in that vat and assemble it into the complex machine that you need. And again, this is on the atomic scale, so you can design things that you cannot design normally from top-down methods, right? And so that really piqued my interest and my imagination as a college student, being able to fabricate things, being able to grow things, I should say. So growing something like a jet engine, that would be an amazing engineering feat. And then I thought about where I was at. So I was in a bioengineering lab, and I realized, you know what? We kind of do that already. We kind of make really complicated things in a vat. And think about a human being growing for 9 months in a vat full of amniotic fluid, right? That is kind of very similar to this whole concept of growing a complex thing in a vat. And I would also hazard that a human being is immensely more complex than a jet engine. You look at the complexities of the human being, and it's just astounding, right? And so the key here for this particular inspiration is cells as micro machine assemblers. So maybe we don't need nanotechnology assemblers made of, synthetic atoms, synthetic materials. Maybe we can just use what nature has already evolved. And if you look inside a cell, you see nanobots, essentially. You see nanomachines that are evolved for very specific assembly and disassembly reactions. On the top left here, this is RNA polymerase taking DNA and making RNA from it. And on the bottom, this is my favorite nanomachine, essentially, which is a ribosome. And this one can take any RNA, read that information, and spit out a growing protein that forms the rest of these nanomachine assemblies, right? And so you have this symphony of different nanomachines inside a cell. And the cell itself can be seen as a micro machine as well. So not on the nanoscale, but on the micro scale. And so you can think of the cell now as a factory to make materials, to assemble things in an ordered way. And so here I'm showing, this is a zygote. This was you at time 0. And that one cell, that you used to be, that you can see under a microscope, that one cell contains all of the information needed to make every single structure, every single biomaterial that is contained in the human body. So think of bone, right? So calcium, phosphate, minerals, seeded onto collagen. So bone is amazing, stronger than steel, right? You have the enamel and teeth. You have optically clear materials, for example, in your cornea that allow light through just the way it needs to be. You have the hair, you have nails, you have obviously the huge amount of extracellular materials such as the collagen and fibers that that basically hold you together in terms of the human body. All kinds of fluids that are made, and all of that is encoded in a single cell, which is absolutely strikingly amazing. And so a cell can do, not only as a factory to make materials, but it can also self-organize that in an amazing way, where everything is where it needs to be for the most part, if everything works well, essentially, right? And going beyond the human body, let's take a look at the rest of nature. We have all kinds of biomaterials in nature. We haven't even we don't even know how to fabricate ourselves yet. That's something we work on. Organ engineering, tissue engineering, that's what we do, right? But everything else in nature around us is astounding in terms of its biomaterials, in terms of its ability to make materials that can be used and utilized. And imagine if we could tap into all of these. So now we're going beyond the human body. We're going into every single living organism on Earth. And that is a huge palette of materials, living materials, or once living materials, I should also add, that we have at our disposal for building literally anything around us. And so a lot of you might notice this figure. This is from the review paper that Martin was talking about. And so again, this is going to a thing that I always gravitate towards, which is the seed and the tree, because it really embodies this principle very well. So on the left is the natural pathway that nature has evolved, where again, you have a single unit, either a single cell, such as a zygote, or a seed, a dormant seed, and it has all the information it needs to build the materials it needs and the structure that it needs. And that cycle repeats itself over and over and over. And key here is the idea of morphogenesis and self-assembly and self-organization, and maintenance and renewal. Okay, and so on the right here is our poor attempts right now at engineering this complexity, this system, where we take a single engineered cell. It can be a bacterial cell, which is the easiest for us right now. It could be fungi, it could be a mammalian cell. And the idea is, do we have the means, the expertise, and the knowledge necessary to go in and tinker with that program right now in that single cell and have it evolve into something much greater than what nature has given us? Can we direct it to build materials that are artificial, that don't exist in nature? And so that is the whole task of living materials for me, from a synthetic biologist's point of view. And then we're going to go into little vignettes of how I kind of explore that in my work as well. But key here is you see this hierarchical organization through different length scales. So from the nano level, which kind of we've been talking about from an abstract level, all the way to the micro scale, to the macro scale, and then finally you get to something that is, beyond macro, I would say, which is an ecological level of living structures, such as entire forests, for example. And so that is the inspiration. Those two themes, self-assembly and cells as organizational factories that we can program and engineer. Those two things were the two kind of key inspiring concepts that I really want to focus on in my work. And that theme runs through a lot of my work. And there is a particular change between vignette 1 and 2 that we'll talk about. So we're going to start off with the vignettes. This is basically a summary of some of the work that I've worked on. And so this work started really in my postdoc. So my postdoc at Harvard School of Engineering in the laboratory of Neil Joshi. And so my graduate studies have primarily focused on protein engineering and synthetic biology. And I really wanted to focus on self-assembly. So I wanted to look at nanomaterials. I wanted that expertise in nanotechnology, nanomaterial synthesis, nanomaterials characterization. And so what we were working on, what I was working on as a postdoc in Neil's laboratory, were these synthetic molecules called DL cyclic peptides. So this is a peptide. The backbone is made of a peptide, but it's chemically synthesized in that you don't have an open start and an open end. You basically take the peptide and you fuse it together into a ring. So now there's no start or end to it. It's just a basic ring. And it's made of these alternating D and L amino acids, okay? So there's a DL cyclic peptide. And these cyclic peptides, they self-assemble into rings that stack on top of each other that are stabilized by hydrogen bonding, right? And so these are, this figure shows 4 different DL cyclic peptides, all with different functional groups shown here in the R, right? Different backbones. And they all stack on top of each other and form these very rigid tubes that are stabilized by hydrogen bonds. So basically the same forces that stabilize ice water or stabilize water in general, right? And so hydrogen bonds are one of the fundamental forces that kind of underpin all biomolecular interactions on the molecular scale, on the atomic scale. And these DL cyclic peptides can form structures up to a micron in size. And incidentally, this whole self-assembly process is very similar to amyloids. And if you guys are familiar with amyloids, you hear about them a lot when you read an article in terms of amyloids being the causative factor it's thought to be the causative factor of Alzheimer's, where you have these Alzheimer's plaques. And what they're made of are these highly stable, hydrogen bonded, stabilized amyloid structures. And so these structures are amyloidogenic, and that's the way they form. And I had gone to a seminar on amyloids, because that's what we were working on at that time, by the late Susan Lindquist. So she's a researcher at MIT. She was a researcher at MIT. That she gave a talk at the Broad Institute, where she talked about amyloids, and she's a pioneer in pathogenic amyloids. So you might have also heard of prions, and prions they replicate using that same mechanism that amyloids do. And so Professor Lindquist was a pioneer for prion disease and exploring prions and how they've evolved. And what she mentioned in her talk was, hey, amyloids are also used functionally by a lot of living organisms, such as bacteria. Bacteria actually make these amyloids, and it's not a toxic form to them. It's not something that is aberrant or something that is, it's actually something that they've evolved to use in a beneficial way for themselves. And so here I'm showing E. coli on the left here. And what you see, those little fragments around, those are curly fibers. And they're called curly fibers because if they get long enough, they actually curl into big curls. And these can be many, many times the size of the actual cell. And what these bacterial cells are doing is they're secreting out these amyloids. And these amyloid curly fibers are acting almost as rebar. So you can think of them as forming the rebar for a highly, highly stable biofilm. So now they have this super stable biofilm that they've made. They've basically secreted this house, if you will, that stabilizes them in a turbulent environment, right? And so this is showing a macroscopic E. coli colony in the middle here. And that colony is formed from a bunch of E. coli cells and curly fibers. And so if you have curly fibers, it actually gives you this macroscopic appearance of all of these invaginations and patterns in the colony. And on the very right here is from our lab back in 2014, where we grew curly. And here you see these cells in pink here. And those curly fibers, you can see it almost forms a honeycomb-like structure, completely encasing the cells. That is in green here. So those are the curly fibers. And so the concept here is, can we take those curly fibers, which are genetically encoded, and it has its own dedicated system that the cell uses to spit out this curly fiber, where it self-assembles on the outside of the cell. You can imagine if it self-assembles on the inside, that would be very bad, because it would gum up all the internal mechanisms of the cell. So it secretes it first as a monomer, and that monomer spontaneously self-assembles to form this huge fiber, which then coalesces into this larger superstructure that can cover like an entire surface. So this is basically a biofilm. It's a biofilm that you see, for example, in the bathtub if you don't clean it, or when you go outside and you look on the outside of any air conditioning unit, there's a huge biofilm. That is, nature's way of having evolved these mechanisms for these bacteria to survive. These bacteria have it's an amazing strategy. And the idea is, can we piggyback on this structure where these bacteria are making their own materials that are self-assembling? So again, this is a very nice microcosm of those two tropes, those two themes that I talked about before. One is self-assembly, the other is a cell as a factory. And here it's actually quite simple because it's a single monomer that is easily genetically tractable that we could engineer. And so the concept is this: we would use that self-assembly domain of curly as the backbone, and every different color here is a self-assembling unit. And you can see it actually has a hierarchical organization to it. So each single protein unit that self-assembles also in it has a motif that is repeated throughout the protein where it also self-assembles through that motif. You have these 5 amyloid domains that stack on top of each other in each single protein, right? And so what we're going to do is we're going to piggyback on top of that and fuse something of interest to that amyloid so that it self-assembles into this long filament and carries along with it our genetically encoded variable function. So this is a function for whatever we want to put into it. And so the larger general concept was, now you have a factory here in the middle. You can input all these molecules, such as sugars, such as key essential factors that it needs to grow and survive. And from all of these different factors, it will spit out for you a material that you dictate, right? And we can control this using synthetic biology, not only what kind of material it is, but when and where to make these materials as well. And then you will have this essentially engineered factory sitting on a surface, which is what we're showing here. And as long as you feed it the right feedstock, it will just continually spit out this material that you have made for it. And what we've shown in our 2014 paper, this is published in Nature Communications, is we can swap out that functional domain and make these filaments do different things. So we can make it template nanoparticles. So this is a gold nanoparticle templated on curly fibers. And so you can't see the curly fibers anymore because they're all surrounded by gold nanoparticles. So this would be a conductive kind of filament that we've just made from scratch. Obviously, you need to add gold in. So each of these materials requires its own specific input to make the material. You could also program the curly to adhere to different materials by adding on a domain that will specifically bind to specific materials as well. And also you can expand this out greatly by immobilizing other proteins to that one filament. So now you can immobilize enzymes that do different things. Here on the bottom, we have immobilized a fluorescent protein, which is the go-to thing to do for synthetic biology to show kind of function. We use fluorescent proteins all the time. And so you can make things that glow, you can make things that catalyze chemical reactions. And so this kind of platform can make a lot of different materials, which is pretty amazing. And it's not only focused on materials that you can only make at the nanoscale. So a lot of the things we're showing here are using SEM or TEM to see these things. So it's on the nanoscale, but you can also make things that are large enough such that you can hold it in your hand. And so what I'm showing here is a video where that red piece of fabric that you see is actually a curly mat where I have decellularized all of the cells. So now we've gotten rid of all of the cells, and all you have left is that curly mat that is composed of fibers. And theoretically, you can make this the size of a table, the size of a house if you wanted. It's just quite expensive. It's not cheap to make these things. So that's something we'll go into in a bit. And in the Joshi lab, they've continued this work since I have left the Joshi lab. And they have, for example, expanded this into something called aquaplastic, where they also have the curly system. You can filter it. And from this filtered material, you can have a living material that still has the cells, or you can have a decellularized non-living material here. And you can make objects out of this. So this is actually a plastic material that you can actually weld using water. So you just take these two materials, you put water, and where the water actually hits these sheets, it'll actually weld it like you would normally solvent weld a piece of plastic. And here you can see that they took these pieces of aquaplastic made of this biofilm. And it's very flexible, and you can actually make a structure from it. This fantastic work by another postdoc in the Joshi lab that took this platform and expanded it significantly. So that's the first vignette, and this work is ongoing. And really quickly, I want to go into what I've also kind of shifted gears. The last couple of years, I've been working in the Collins Laboratory, also at the Wyss Institute. So Neil Joshi's lab went to Northeastern. I stayed at the Wyss Institute, and I stayed on in the Collins Laboratory. And the work that I did now for the past couple of years is actually moving away from living materials. And the reason I did that was a lot of the responses that I got from living materials was concern about the gray goo scenario for living materials. So you remember the gray goo is like, what if it runs amok and we can't control it and it destroys the environment, right? So I had a lot of concern related to living materials from that kind of perspective as well. There's a lot of biocontainment concerns, which is, so when we published a lot of our papers and went to reviewers, they're like, is this safe to have genetically engineered materials that can regenerate and grow themselves? That sounds great, but I'm not sure we would want that. And so we would always have to have this option of, here's an example where we killed everything. We just made the material, but then we killed the living thing in it. So don't worry, it's safe now. And so, to me, that's not that, we had to do that for a lot of these papers, just to show that there is that possibility. But you really lose, to me, what is that magical ability, which is the ability to generate and the ability to grow from your surroundings. That ability for that crystal to kind of have a living program that can take from its environment and build something from scratch, right? So what I wanted to explore in the Collins lab was something that they had worked on just as I was joining, which is the cell-free technology, which is, can we take synthetic biology, everything we've learned and everything we're able to engineer using synthetic biology, and use it in everyday technologies without living cells? So again, that bears repeating. Can we do biology without cells? It sounds counterintuitive, but you actually can. And the way to do that is by using something called cell-free technology. You can take cells, this is, for example, your favorite E. coli cell, or basically any cell will work. You basically lyse the cell, you lyse open the cell, you burst open the cell, and you collect all of the machinery, all of those self-assembling protein nanofactories that we talked about earlier, and you isolate those, and those are now a cell-free extract, okay? And so those contain all of the machinery inside the cell, and they're actually still active, and they're actually still functional. And so what people do, and people have done this for decades, this is since the 1950s, people have done this, is they've taken these cell-free extracts, which are basically extracted machinery from inside the cell. You can add nucleotides, amino acids. So basically all these additives are things that a cell would normally need to survive. So all of the inputs, essentially, carbon sources, metabolites. You can add in your circuit, which is now encoded on a plasmid or a piece of DNA. So this is the design part. This is what you design. So this is the program, your genetic circuit, and this is the chassis, the engine that you're feeding your program into, or the computer, if you will. And in the end, you get the cell-free reaction. You mix all these together, and this reaction will actually mimic living cells until it runs out of these energy sources. It will, for example, if you have a program in it to make alcohol, it will generate alcohol and breathe out carbon dioxide, but it's no longer living. It's just kind of a zombie reaction of what was once living, right? And so this is quite powerful. And what the Collins lab found was you can actually take these cell-free reactions and you can freeze dry them, okay? And you can actually take them, freeze dry them, and as a freeze-dried powder, it's actually shelf stable. You can put it on the shelf. You don't need to refrigerate it. So unlike any other living cell, just like your leftovers, you need to refrigerate it or it will go bad. So what they found was this lyophilized cell-free extract can be, stabilized at room temperature. You can transport at room temperature. You can throw it in your drawer and come back to it in a year, and it would still be good. And all you need to do is add water to it, and this entire process will pick up off where you left it before you freeze dried it. So this program that you put in, if you put in, I want a program where I can make a fluorescent protein and have that reaction glow. You add water to it, it will happen. After sitting on the shelf at room temperature for months, which is shocking, right? And so with this technology, there was a huge amount of this opens the field for all kinds of different technology now, where we can apply synthetic biology, which before it was always complicated by the fact that you needed to keep living things alive. So the synthetic biology technology is very different than the technology we have at home. For example, your cell phone or your iPhone, things that you use every day. You don't have to keep those things alive. You don't have to feed those things. But in anything that is a biological technology, you do. And so what we've basically found is a way to put a pause on biology and have that technology on demand. So now anytime you need to access it, you want to execute the program, you just add water and the cell-free reaction will boot itself up. And the analogy I like to make is very much like ramen. So ramen technology encapsulates this cell-free technology very well, where you have something that's very shelf stable, stable for years, if not decades. You have all the parts that are stabilized separately, and all you have to do is mix them together and add a little bit of water. And then in the end, you have something that, a priori, you would not imagine you could make and store in such a stabilized format, such as soup, right? And so using the cell-free technology, we apply that to a number of different things. And so for anything in synthetic biology, the first couple of things that you normally apply it to, my interest is materials, material science and materials research. But the first couple of things is always diagnostics or therapeutics, because that's usually where the money is. That's where the interest is in terms of publications as well. And so you're forced to kind of do that first. And so using this technology, the Collins Lab was able to put those reactions on a piece of paper and have these diagnostics that just change color when you expose it to different things in the environment. For example, Ebola, this will change from yellow to red in the presence of Ebola. And again, this is a piece of paper with this technology embedded on the paper and freeze dried into it. That's the size of a stamp, right? And so that's something that normally would take a laboratory to do. You can have in the palm of your hand or in your pocket now, right? And you can already see the possibilities of this for material science. So not quite living materials, but something along the lines of non-living materials that is biologically generated. So just to skip ahead, we published a number of papers on different things, and we ended up gravitating towards wearable synthetic biology. So we're finally getting to the materials realm of my own interest. And in specific, we're not talking about bulk materials, we're talking about textiles here. So anything wearable, such as textiles. And I'm showing here two pieces of work from the Liu lab, and this is from the Wang lab at MIT as well, where they've actually put living cells in wearables. And so this is what a lot of people have done. They've just taken what we've been able to engineer in synthetic biology, which is living cells that have some kind of programmed circuit in them, and they've fabricated some kind of textile or flexible substrate that you can stick those living things into. And you have living cells now that you can wear on you that will detect something. And here on the right, you have living cells embedded into this textile here, and the cells will actually open or close pores in the textile depending on the amount of moisture that's in the atmosphere. So due to relative humidity, your shirt that you wear will open up and allow more air in, and it'll automatically close back up when the humidity drops. So it's a very interesting way of using living cells in materials. So one criticism of these technologies, which we found absolutely inspiring and mind-blowing, this is a great example of living materials that is a bit more intimate than what we normally talk about when we think about living materials, which is, normally people think about buildings and things of that sort, or nanomaterials, those two length scales. This one is kind of in the middle length scale, something personal, what I like to call personal technology, things that you use that are very close to your body. How do we integrate living things into them? So now these are essentially engineered second skins. And one of the criticisms of these technologies that we came across was that living cells that are engineered should not be put into anything that close to you. So any wearables. And so there was a push to actually engineer the complex synthetic biology circuits, but have no living materials, which is what we applied the lyophilized cell-free system to. So this is the concept that we came up with, which is wearable synthetic biology using these non-living materials now, having these freeze-dried cell-free reactions. And anytime you are hit with water, they will actually rehydrate and form some kind of chemical reaction. And so this combines two things now: synthetic biology, which is interfacing with living systems, complex functions, and the ability now to be programmed. Again, going back to the whole idea of programmable cellular factories. And now we're actually getting rid of the factories. We're getting rid of the self-regeneration, self-renewal, which is actually, to me, the most powerful part of that technology. But for the short term, to alleviate these gray goo concerns that a lot of people have been having, we wanted to integrate into something intimate, such as wearable technology, highly portable, personal technology. And the easy way to do that for us, the way we saw, was to use cell-free extracts and DNA, embed that into textiles. So now you have the programmability, the versatility of living systems without necessarily having to worry about biocontainment or anything self-replicating, because you no longer have a living cell. You just have the machinery, the engine inside the cells at your disposal now. And here is what we ended up designing. This work was just published last Monday, in Nature Biotech, and the work is a lot larger than what I'm going to go into here, but you guys can check it out. We designed textiles that have the synthetic biology circuits that will immediately respond to anything in the environment that is aqueous. For example, if you are splashed with blood that has Ebola virus in it, the fabric will be able to change color from yellow here to purple. So you have actually now a textile that can be very large, and it's embedded with synthetic biology to change color to respond to something on the molecular scale. So it will know whether or not you were splashed with something that has Ebola or Zika or some other virus, for example. And we also have made a textile where we embedded it with fiber optics. And the fiber optics allow you to actually detect light coming from these reactions. Now we can make reactions with these cell-free reactions that are freeze-dried. Here we're adding viral RNA. This is HIV. And you can see the reaction will actually generate light when you have HIV RNA around. And that light can be detected by fiber optics. It gets connected to a portable detection unit, and you can now have a system where it's constantly monitoring you from synthetic biology circuits to actually look at what you're being exposed to. And probably what has gotten the most press is what we developed next. So obviously last year was the year of COVID-19. We took that programmable synthetic biology textile and incorporated it into face masks. And what we were able to do is have a face mask that can actually amplify and detect COVID-19 nucleic acids from your breath. So you breathe on the inside of the mask. the mask itself, it has no instrumentation. There's no pump, there's no electronics, there's no battery. Everything is actually completely integrated into the material itself. So it's once it hits the material and you press a button, and that button activates this reservoir of water here. Like anything that's biological, you need water for it to happen. That's all you do is you activate that blister of water. That water flows through the sensor and basically will amplify and detect the presence of COVID-19. And it's able to do this quickly. So we can do it in about 90 minutes for a nucleic acid amplification test. That's actually really fast. At the point of care, you don't need to go to a laboratory because we've basically taken everything in the laboratory and reduced it down to a piece of textile that you can hold in the palm of your hand. And the sensitivities we get are on par with the WHO approved RT-PCR level results, assays for detecting COVID-19. So this is the gold standard, where we hit the gold standard in terms of a laboratory level detection system that it works at room temperature and requires no laboratory. And it's also highly specific for SARS-CoV-2, the COVID-19 virus, compared to other coronaviruses. So this is, this really shows you how that one concept of taking synthetic biology, the best things out of synthetic biology, cutting away what people are concerned about, which is the ability to self-replicate, the ability to grow, beyond our own control, and just take the programmability part and embed it in textiles. And so I'm going to end our vignettes here, but next I'm really interested in taking this further for other materials besides textiles. So you can imagine this as kind of this neutral zone between living materials and completely synthetic materials, where you have biohybrid materials, where you don't have that biocontainment concern, but where you use the programmability. And now you can add this into textiles, you can add this into bulk materials. And I'm kind of excited to see where we're going to go next with this. And that's what I'm working on now. So with that, I want to close this with just a quick look at some challenges and opportunities that I think the living materials community, should really look at. And these are challenges and opportunities. You might have your own. These are particular ones that I focused on. One is, in the past couple of years, this whole looking at synthetic meat. I know it's kind of odd to talk about this with living materials, but I think the trajectory of impossible meat or vegan meat is great in terms of what it portends for living materials, what it portends for taking living materials and taking it out of the lab and into commercialization space, where it can actually enter everyday technology, right? And so if you look at impossible foods or this kind of fabricated meat, a lot of the technology is actually from academia. So they've basically pioneered the process of taking plant-sourced heme, for example, and integrating that into other plant-based fibers to make something that is very similar to meat. So this is their factory. You can see it looks nothing like a meat processing plant. This looks like a bioproduction facility because that's what it basically is, right? And the lesson to learn here from this particular example is the ability for them, for so long, vegan meat has been around for decades. Tofu burgers have been around. But in the past couple of years, you've seen such a huge spike in interest and a huge spike in investment. And a lot of big names are signing on. You have outlets like Burger King that are commercializing this and going crazy on social media and things like that. And so we're coming to this point, this inflection point, where this technology that has floundered for so long has catalyzed and now has entered the mainstream. And the lesson to learn here, I think, comes from the founder, Patrick Brown. And I just want to read this real quick. The food industry is decades behind the times. The stuff we're doing now that's new to the food system was old news 40 years ago in the biotech world. And so Patrick Brown was actually a professor at Stanford. He worked in DNA, microarray technology, and cancer biology. And his whole Impossible Burger idea came up during a sabbatical, where he thought about adding hemoglobin into plant substrates. So the ability to take something in synthetic biology and apply it to industries that haven't really thought about using biotechnology in that way before in its disruptive manner, I think is a key thing for us to think about for living materials as well. Especially, we're seeing things now. This is, again, all these examples are going to go through the same thing, which is changing consumer sentiment. There's now a huge consumer sentiment at not leaving a footprint anymore in terms of how we impact the world, much more so than there was 10 years ago. And I think piggybacking on that sentiment of that need, of that want for the market, there's an actual market desire now for these kinds of technologies. So the opportunity here is to learn from related industries. What can we learn from the food industry? Because I think it's actually the closest to living materials in terms of the production and the distribution. Just in the case of food industries, you eat the material, you don't, use it to build a house and live in. Which brings us to our next example, just really quickly. Evocative Design. So this is fungi, and a lot of you know about this as well. IKEA has already signed on to this. This is using a fungi as a bulk material for packaging, and they've also used it for leather and also bacon. They're going into the food route. And this also came from a different area as well. So this is art. So a lot of people don't know that this fungal technology was actually used in mushroom art for the longest time. So the lesson here is look into other areas as well. So I think the exploration of art and living materials is going to have a profound impact on commercialization and exploration of these kinds of technologies, not in the lab, but look to artists, where artists are outside of the lab, they're thinking outside of the box. And I think that's going to be the fruitful area to focus on. And the last thing that I want to focus on, and then on the top there, is upending disruptive technologies that living materials can piggyback on. And one thing that I clearly see right now is 3D printed fabricated houses, 3D printed buildings. So can we piggyback on this already disruptive technology that is already disrupting the industry in terms of fabricating living spaces, and somehow integrate living materials into that? And I think living materials is poised. It's much easier to integrate living materials in the way that these layer by layer assembly methods are being implemented right now, rather than traditional construction methods where you would have substantial pushback in integrating living materials, I think. Sorry for the rushed ending, but I'll finish right there. And with that, I'll leave it open for any quick questions.
Thank you very much, Peter. Sorry, I'm just appearing out of the blue there. Thank you so much. I particularly welcome the final comments about disrupting industries, and I'm also glad we got our robot arm now, because we were thinking about doing extrusion type work as well. So there's an awful lot to talk about there. We're kind of out of time for this session, so we're kind of into the break and the prep before the next session. But rather than do it this way, because there are quite a few questions in the YouTube chat. So Peter, would you be able to jump onto the YouTube chat straight after this session and maybe answer those kind of live in the chat boxes? Would that be possible?
Sure, absolutely.
What I'll do is I'll share with you, as I have done with the others, the link to the YouTube chat. So that's just in the chat box in Zoom here. So if you click in the chat box in Zoom, you'll take you to the chat in YouTube, if that makes any sense at all. There's an awful lot of interest around the cell-free systems that you were talking about as well. It's something that we've thought about a little bit, but haven't really gotten our heads into yet. So that it would be really good if you could continue that discussion.